Hello everyone, this is our next video. We are going to be doing the lesson Cell, the fundamental unit of life. Alright, so when we were in class 8, there was one chapter called Cell Structure and Function. Yes, there was a chapter called Cell Structure and Function. In this chapter, we learned about cells, classification of cells on various bases and structure of cells and cell division. Now, today we will be doing the same thing, except we are going to learn each thing in a much more advanced manner in class 9. Okay, fine. Now, cell. What is cell? Now, cell is basically the structural and functional unit of life of every organism. Okay. So let me just draw a simple diagram. Okay. Fine. So basically, let's say this entire thing forms a tissue. Okay, now let's say I'll make you understand this in a much simpler manner. Let's say we assume each of this is a piece of Lego. Now we are constructing a Lego wall. Fine. You are constructing Lego wall. So if you use only this block, would you be able to make a wall? No. You have to keep on joining, keep on joining, keep on joining. That is what is a cell. Now, similarly, if we take each one of this hexagonal structure to be a cell, we can form out the levels of organization. So let us put the heading. Levels of organization. Okay, the levels of organization in a living organism are as follows. Fine. Now, first we have cells. Now that we have already assumed how our cell looks like, so this is our cell. Fine. We could just put a small dot like this in the middle. Will show the nucleus. What is nucleus? I'll explain it later. Okay. So this is our cell. Fine, cell. Now, when I have these cells and I join them together, okay? Goodness gracious. Sorry guys, I'm just wasting time. Okay. Fine, okay, this. Now I assumed each hexagonal unit as a cell, right? Okay. When we assume each hexagonal unit as a cell, okay. Fine. Now you see, like the Lego. You join the Lego, you create a wall. Similarly, each cell plus cell plus cell plus cell plus cell plus cell, like there will be many cells, there will be a large number of cells. All of these cells together will form one thing called as a tissue. So this is called as a tissue. Fine. Let's say now I will duplicate this. Okay. I will duplicate this 1, 2, 3, 4, 5, 6. So let's say I'll pause it. Okay. Now see this is my one group of tissue. Okay. This is my, see, a tissue will contain a large number of cells, but just for example. So this is my one group. This is my second group. Let's say I name this G1, I'll name this G2. And this is, both of them are the same tissue. Now these two together will join together to form something. Now what is that something called? This is called as an organ. Fine. But these two tissues are not of the same type. Okay, I'm just saying that the same number. So we form an organ. Okay, so I'll take this as T1 and T2. Okay. Now, wait. So pause. Okay. So now here let's. So this is our 12 month. We have 24 tissues. Sorry, we have 24 cells. But this is, we know, is our organ 1, which is different. This is our organ 2, which is also different. So these two organs do something related to each other, do a function that's related to each other. Like I said, help in digestion, in different processes in digestion. So they together form a system. What is the system called? Digestive system. Fine. Okay. Now, many of these organ systems together form an many organ systems together form an organism. Fine. So if you look at what we have just studied, we have studied that cell is the structural and functional unit of life of every living organism. We have studied about the levels of organization in different living organisms. We have a cell, then we have a tissue, we have an organ, we have an organ system. Now what can we infer from this? We see that cell is the starting point / the lowest level. Okay. Now from this we can infer that same cells perform a specific function to form a tissue. This one, different tissues join together to form an organ. Now these tissues, these different organs together, different organs form groups which form an organ system. Different organ systems together form an organism. Fine. Okay, so this is the levels. These are the levels of organization within a living organism. Okay, so if we depict this now, we have cell, then we have tissue, then we have organ, then we have organ system, and then we have our organism. Fine. This is the level. No, these are the levels of organization. Fine. Now, let us study a little bit of history. Fine. Okay. Fine. Now, first, we need to learn about how this history had happened. The main reason about this history is linked to the discovery of cells and the discovery of microscope. Fine. Now, if you have with the new syllabus here in the corner, they said we need to know about recent advancements in cell biology, which is basically about the types of microscopes and all of this. So that's what we will also be learning. Now, cell discovery, okay, is linked to the discovery of the microscope. Fine. It is linked to the discovery of the microscope. Now, what is this microscope? Microscope is a device. Okay, fine, let's define it properly. High resolution instrument used to see very small objects. Fine. Now, first let us learn about who invented our microscope. Who invented it? A person named Zacharias Janssen. Alright. Zacharias Janssen also invented the microscope in what year? 1590. Okay. Now, just like the Right to Information Act or whatever acts in SST you learn, they will be revised. In the same way, this Zacharias Janssen's model was also revised. By whom? It was revised by Galileo. You know who's Galileo, no? In 1610. Okay. Then later it went to Robert Hooke. Fine. He went to Robert Hooke. So, what have we studied? We studied about cell discovery is related to the discovery of the microscope. What are microscopes? Microscopes are high resolution instruments used to see high resolution instruments that are used to see very small objects like cells, for instance. Okay. Who discovered the microscope? Zacharias Janssen. Remember the year. Then we have Galileo. Fine. Then we have Robert Hooke. Okay. We have Robert Hooke. Now, this person called Robert Hooke was the person who discovered the cell. He discovered it in 1665. Okay. So basically, Robert Hooke invented his own microscope. Fine. So he invented a microscope. Okay. So you see, right? He discovered it in 1665. Remember the years? These are very important. Robert Hooke had invented his own microscope. Now, using this microscope, he discovered the cell. How? See, he took cells he took a piece of cork. Okay, he took basically this expands into a full piece of cork. Fine, a small slice. And then put under a microscope. Fine. So he absorbed it. Now what did he see? He absorbed this. Okay. See, he observed many holes. But then here we are talking about something at a microscopic level. And then they are called as compartments. Now, these compartments were partitioned. There was a partition over here, and they're partitioned horizontally. This is just one row I made. There are many rows. Hundreds and hundreds of compartments inside the one slice of cork. Now, what did he name these substances? He named these substances called cells. Now, of course, Latin is a great language. Basically, almost every scientific term is derived from it, like acid or whatever. Okay, so that is why this word cells was derived from the Latin word cella. Cella basically meant hollow space or compartments. Fine. So this is cella. Fine. Cella. Okay. So that's what the word was derived from. Now, later it was understood that what Hooke had observed was cork. That was true. But the cork had dead cells. How? See, let's say this line is your tree. Okay, you have this layer. Let's say this is your tree. Fine. You take only this side. This layer is your bark. Fine. This layer is your bark. So this layer stays alive only when it's within the tree. But then what is cork? Cork is basically, he said that he cut it off and sliced it. Fine, cork is made from this, cut it off, and then after that he get that piece of cork and then he sliced it. So this is the piece of cork. He sliced it up. He placed it under the fine, he placed it under the microscope. Okay. Assuming there is microscope, I'd also look at it like this. But then this is how a microscope actually looks. As far as we know. I know, sorry. Not we. So he placed it under a microscope. Let's slice the cork. Okay. Now, when its main source of energy, which is the tree, is cut off, so it's basically dead. So all the cells were dead. He had observed dead cells which became clear. Later, not many years after, so this happened in 1665. The next incident happened in 1674. So it's not really that far away. It's just 9 years. I mean, even though 9 years is a lot for us when we observe it in real time, but then it's not like really long in that time, especially with that technology. So 1665 we saw that Robert Hooke had observed dead cells. Later in 1674 came another Hooke. Okay, not making fun, just so that you guys can memorize. Came another Hooke. His name was Anton van Leeuwenhoek. Fine, his name was Anton van Leeuwenhoek. Who was Anton van Leeuwenhoek? He was a Dutch draper. Draper means, the one who arranges clothes or takes measurement of clothes. I believe that is the meaning in this case. So he was a Dutch draper. Fine, he was a Dutch draper. Okay. Now, so this Anton van Leeuwenhoek was the first to sketch, observe, and to describe. First, this observe had happened, then describe, then sketch. Describe living cells. He described about bacteria, erythrocytes, sperms, and protozoans. Fine. Okay. So what did he do? How did he observe this? So it's a simple experiment. You would easily understand this. So like, let's say you have a beaker. Leeuwenhoek's experiment was the same, but then he didn't use a beaker most probably, because beakers most probably did not exist at the time, or it's me stupid at history. Okay. Now let's say this is the beaker. You must have noticed that stagnant water pretty much just you have many mosquitoes breeding in them, right? So that's what he took. He took water, he left it, fine. Then later he took a sample of this water, maybe either using a syringe, but I'm showing how you could do it at home. So he took a sample of this water and put it under a microscope. Okay. So I put it under a microscope. What he had observed was that he saw bacteria, and he conducted several other experiments. Also, he observed substances moving inside, and he called these substances as animalcules or animalcules. Fine. Okay. So this is what Leeuwenhoek had done. Now let's have a recap. We have a first person called Robert Hooke, who had invented his own microscope. He had observed cells. He had observed that they were divided into they were many compartments which were partitioned out. He called these compartments as cells. Then later many people had understood that he had observed dead cells. Then later a person came called Anton van Leeuwenhoek. He was a Dutch draper, and in 1674 he had discovered living cells by performing the stagnant water experiment. And he observed several new discoveries, and later came a person who gave a cell concept. So there was a person who knew about these discoveries, and later again, not really far away, but then just okay, I'll just tell you this. We have a person named N. Gru, which you may have not heard of a lot, but then he still gave a concept in 1682. Okay, so till here we have 1682. Then later we have a big jump. Okay, so like you have a big jump of 150 years from now, from this point. So what did Engru say? He proposed to biology. It's like, biology, I love you. And then he proposed a concept. Fine, he proposed a concept that cells are the structural and the functional unit of life. Okay, structural and functional unit of life. Okay. So basically, he just gave a concept which we have learned before, which is gonna be so boring. Okay, about this thing, you'll be hearing the same thing almost at least 10 to 15 times in this chapter. But then this chapter is not boring. Okay, so let's just take a recap from the first. Cells, structure and function. We learned about all of this in 8th. Cell, what is cell? It is a structural and functional unit of life. The levels of organization in living organism are as follows. Then he gave this thing. Then I drew this thing, a hexagonal unit which is a cell. Cells form tissues, tissues form organs, organs form organ systems, and each organ system forms an organism, different organ systems. Now, we have microscope, discovery of microscope. So the cell discovery and microscope discovery are related because microscopes are used to see cells. Zacharias Jonassen discovered microscopes in 1590. Then later we have Galileo in 1610. Then Robert Hooke, who modified Zacharias' microscope. Then Robert Hooke discovered the cell. Then later he observed it in cocks. Then it was found out that he had observed cells which were dead, not alive. Then we have a person named Antony van Leeuwenhoek. Okay, we have Anton van Leeuwenhoek. He was a Dutch draper. He wanted to achieve something, and so he he was the first person to observe, describe, and to sketch living cells like bacteria, erythrocytes, sperms, and protozoans. And Leeuwenhoek had basically performed this experiment, which had helped him out in life. Then so basically Leeuwenhoek had successfully made it into our NCERT textbook around about 400 years later, I should say. 300 300's too low. 351 years later, And so later we have another person named Ian Guru who also wanted to get into many books but then got into a few books and did not get into our NCERT book. I believe he did, I don't know. Maybe in the new book. He proposed a concept that cells are the structural and functional unit of life. Okay? So, after N Guru, we have a 150-year time lapse. And nothing, absolutely nothing was known about cells except for everyone relied on these discoveries because there were no powerful microscopes. All the microscopes were normal. You basically had a normal lens converging microscope, but then you did not have those compound and light microscopes that you use in laboratories. Okay, electron microscopes are too high. They were discovered like around 1932. So you can't say electron microscope, at least a compound and light microscope, no. So around 150 years people were trying, but then there was no progress until there's no progress. So like maybe at least not something major, but there was a progress, but it was not really that major. Played a major role in this. So after N Guru, we have a person. His name was Robert Brown. Okay. See, Robert Brown, I believe, thought this way, or at least maybe you could remember this. So it happened in 1831. Robert Brown must have read a lot about Robert Hooke's discoveries. Must have read micrographia. Now, what is micrographia? See, you need records / manuscripts so that you can know. So, like now, if you see Robert Hooke had written a book, had explained all his observations in micrographia. So he must have read Robert Hooke's book of micrographia, and he must have thought so Robert Hooke had Robert in his name, so he became famous. His parents would have read micrographia and be like, Robert Hooke had Robert in his name, so he got famous. If I name my son Robert as well, he'll get famous. Actually, this is true. If you look at Robert Hooke, he got famous. Robert Brown, he got famous. Then we have our Iron Man, Robert Downey Jr., who also has Robert in his name. Okay, so like just think of this. Okay, so he read this and he might have probably got inspired, and then so he discovered the nucleus. Fine, he discovered the nucleus. Now, from where did he discover the nucleus is the question. He discovered the nucleus from the root cells of the orchid plant. Okay, so there is an orchid plant, there were the root cells, and from that he discovered the nucleus. Now, we will learn about what is nucleus when we are learning structure of the cell. Okay, now this is what Robert then discovered the nucleus. Next, after him, we had 3 people, but different years, who worked on the same thing. First, we have Sheldon. Fine, Sheldon from 1838. Matthias Sheldon from 1838. Then we have Swan from 1839. Okay, so between these two there was only a 1 year gap. But then later came one more person called Virchow, Rudolf Virchow, from 1855. Now, all these people had one thing in common. They were Germans, fine, which is something that is used to define their native place. Okay, so they were Germans. Okay, they were Germans. These two were German biologists. Biologists. He was a German physiologist. Alright, okay, now. Okay. Now, if you look at Germans, if you watch, I like chess. So if you have watched the channel Gotham Chess, you could have, and you watch him almost all his videos. There was a point when he said that Germans play chess like they built BMWs, and that accounts for not only for the chess sector, but that accounts for academics. So like Germans studied the so precise, like the way each part of the BMW is joined and you get the satisfying sound. The same way they study in such a detailed manner. Alright? So this is. Now, what did all of these Saint Germans do? What did all of these people do? They proposed something called as the cell theory. Cell theory. Fine. Now, what is the cell theory? See, these people had proposed the following points, and Virchow has a very special contribution. So, they have mentioned these. All living organisms are composed of cells and their products. Second thing, all cells are similar in chemical composition and metabolic activities. Okay. All. Let me think. Okay. So yeah. All cells. Sorry. The functions of an organism are a result of the functions of an organism. Okay, I'm just doing something, okay? See, the functions of an organism are results slash out. No, what am I doing? Slash outcome of the interactions between the constituent cells. Okay, so all living organisms are composed of cells and their products. All cells are similar in chemical composition and metabolic activity. It's a function of an organism or a result slash outcome of the interaction between the constituent cells. Okay. So if we have this and this, okay. Now there was one point that Robert Brown had added. Okay. See, not Robert Brown. Virchow. He was German, so he gave this policy in German. Omnis cellula e cellula, which basically means that new cells are formed from division in pre-existing cells. Fine. So, new cells. Okay, how many pages have we covered? Only 2. Oh, no, wait. Okay, so 3, but then not a lot. So, new cells are formed from division in pre-existing cells. So, he added that point. And therefore, this is what we have today. Fine. So this is what we have today about cells. So basically, this was the updated cell theory. There's also, you can also add one more point, which is basically that cells are the basic structural and the functional unit of life. So after all of this, we have our last scientists, whose names are Knoll and Ruska. Fine, Knoll and Ruska. In what period? During the period of 1932. Now, what did Knoll and Ruska do? They were Germans. Fine. Okay, they were from Germany. They invented the electron microscope. Fine, they invented the electron microscope. So this electron microscope was employed to be used for studying cell organelles. Fine. Okay. Now, after Knoll and Ruska, we have finished the discoveries. Now we have to study about the types of microscopes. Find the types of microscopes. Okay, so there are mainly two types of microscopes. So microscope, compound or light microscope, and electron microscope. Okay, fine. These are the two types of microscopes. Now, what is a compound microscope? Now, a compound microscope uses more than some use one lens, but then compound microscopes, most of them have high magnification powers. Okay, from 300 to 1500 is our range. So that's why they use more than one lens. See, what happens when you use more than one lens? See, you learn, no? Let's say I have a convex lens. Okay. See, I have a convex lens. Then I have another convex lens. See, I have two light rays. Okay, coming from an object. Okay, so let's say this has two light sources, one over here. Okay, now this is an object. It has one laser over here, one laser over here, and you're getting two light. Now, see, this convex lens, what does it do? It'll converge it to some extent. Okay, let's say it'll converge it like this. Fine. Now, see, you see the organism is so small, no? Let's say you have, okay, now I'll just move this here. I'll move this here. Now let's say we have our specimen. Specimen is basically an object. So this is our, if I flip this upside down, you will see it. But then I'm just showing it. This is completely horizontal. This is our stand. Okay, this is our stand. Alright. Now we have a water droplet that is taken from a river, an impure. Okay, so like let's say this is a water droplet taken from a glass of water that is mixed with soil. Now see, this converges. Okay, converge. I am taking physics class for you for a few minutes. This also passes here. Now what will the convex lens do to this ray? This ray will also converge more. No, it will go and hit the exact center. Okay. Okay, how am I supposed to draw this? See, let's say the object is here. I will draw the object with it. Now this will also converge. Okay. This will also converge. Okay. Okay, let's say these objects converge over here. Okay, this fine. So I'll just erase all of this. Fine. And then I'll change this to stroke eraser. Sorry, area eraser. And then I'll erase this entire thing. Okay, I'll erase this and I'll draw the object here. This is your water droplet. This is your stand. Okay. Now it goes and hits the exact center. Now at the exact center you see the light falls over there. So you basically see what is happening inside. The convergence will produce a big image like a magnifying lens and all that. No, so you would basically see all the microorganisms. Okay, all the microorganisms. Okay, and then all those the bacteria, everything that are present in, and all of them you'd see. Fine, I'll change this to area stroke again so that I don't need to. Okay, so that is how a compound microscope works. So it has two lenses. This is the eye. This is a lens that is used for magnification. There'll be a small like transparent glass, or maybe you could also use a lens over there. That lens is called as an eyepiece lens. Now let's say I am using two lenses. If the eyepiece lens is not a converging lens, then you could have two lenses as your objective lenses. So these two lenses are called as the objective piece or the objective lenses. Okay, now there will be certain things which help to increase and decrease the thickness. If it's a small thin lens, you can stretch it out, but then it's not like our eye. But you can change the position of the stage. See, the stage is there, no? In a compound microscope you have knobs, one big knob, one small knob. Now what does the big knob do? The bigger knob, this one, no? You use it for, if you are in a hurry, you use it bigger knob for coarse adjustments. So for example, you need precise focusing, you use the bigger knob for rapid adjustments. For the smaller knob, it's like for fine adjustments so that you can get a precise image. Okay. Now you could also increase the magnification powers like 5x, 10x, 15x, and all of that. Okay. So now let us also recap these types of microscope from here. So now then, discovered from 1932, and also they were Germans, and they discovered a microscope called the electron microscope. It is employed to be used for studying cell organelles. Then we have types of microscope, compound microscope, and electron microscope. First, we are learning about compound microscope. A compound microscope has more than one lens. Either it can have one lens or more than one lens. Okay, 300 to 1500 times it would also magnify. Then, so these two focus it and all of that we have. Now, after all of this, we have an electron. I'll be ending the video soon. This will be part 1 because there is a lot to cover. Fine. Okay. And if you think this is complicated, this is basically not your NCERT syllabus. Now there is this book close to your NCERT called as the Foundation Course by Mtg. That foundation course will be following because most of the 9th graders didn't get your books. Even I didn't get my books, textbooks. So I'm studying from this book and then I'm telling you. Now electron microscopes. See, electron microscopes. Remember there was this experiment called as the cathode ray experiment, which is performed by J.J. Thomson. I believe J.J. Thomson, right? Yeah. Cathode ray experiment. Fine. So basically that's the same thing that's happening in the electron microscope. You use an anode, I think so. I didn't study the structure, but then I didn't study the diagram, but then when you look at if you look at the theory, the electron microscope shoots an electron beam. Fine. It shoots an electron beam. Now see, this electron microscope has several detectors. Now the electron beam will go precisely hit one part. Okay. The electron detector will detect. Now the electron detector is more powerful than a lens. It has lenses as well. The electron detector is more powerful than. Now see, when electrons flowing basically means it's kinda like an electric current. Now electromagnet. See, there's also an electromagnet lens. Electromagnet below the lens or an electromagnet lens as well surrounding the lens. See, as the electron goes, the electron also becomes a little bit magnetic. So after it goes and returns back from the detector, it basically, I think, the electromagnet also sees it and then it uses it for precise focus. So that is what is an electron. Now this electron microscope is somewhere when you compare it to a compound microscope. If the compound microscope, see, let's say if we have this as the electron microscope, a compound microscope would somewhere over here, because you know the magnification of an electron microscope. An electron microscope has a magnification of about, it can range from a lot. It can also start from 50,000, then it can go to 100,000 and all of that. It can go to 100,000. It can range from 50,000. It's completely okay. So basically, it uses an electron beam and detectors everything so that it can find out. So that is what. Alright, so I'll just end the video here and we'll cover everything in the next class. Thank you guys for watching. Please like, share and subscribe. Bye.